seven-membered heterocyclic ring, that is azepine. See, up till now in the same chapter, that is heterocyclic compounds, we have studied five-membered heterocyclic ring containing one heteroatom. So the examples of those compounds are furan, pyrrole, and thiophene. This we have discussed in case of unit number 3. In unit 4, we are discussing, we have started a discussion on heterocyclic compounds containing five-membered heterocyclic compound containing two heteroatom in its structure. So in that we have discussed pyrazole, oxazole, thiazole. These many compounds we have discussed. In the same chapter yesterday we have started a discussion on six-membered heterocyclic ring, that is pyridine. Related to pyridine, we have discussed his importance in the medicinal point of view. It is having so much importance in our medicinal chemistry and in therapeutic index. Then a very important property related to pyridine, we have discussed its basicity. So the question is there 100% on the basis of uh, basicity pattern, aromaticity pattern of the pyridine. And the focus of today's lecture is we are moving towards a compound containing seven-membered ring containing one heteroatom. See, the ring is seven-membered, that is the cycloheptane. It is unsaturated, and one of the carbon atom of this seven-membered ring, it is replaced with a nitrogen atom. So the name of the ring is azepine. See, if we are replacing one of the carbon atom or seven-membered carbocyclic ring, it is known as azepine. We can write the structure of azepine in this manner also, that is in the chair and boat conformation. Now we all know, or you may have studied the different conformations of the cycloalkanes. So this azepine is also coming under the category of cycloalkane family. So with that we can write the structure of all those cycloalkanes in both the conformations, that is chair and boat. He had so many conformations depending upon the degree of their structural deviation. But these two are the stable conformations of these azepines, that is chair and boat conformation. So the boat conformation with the 22% of chair conformation. Now, whether the structure of azepine, it is aromatic or not, that we will discuss in coming slide. So the structure of azepine is planar in nature. The nitrogen atom which is there in the structure of azepine is sp2 hybridized. See, they have the potential of 8 electron systems. Up till now we have seen the 6 pi electron systems. Yesterday we have seen the pyridine. It's 6 pi electrons which are inside the ring. They are taking part in the cloud of aromaticity pattern. But the structure of azepine have already 3 pi bonds, that means 6, and his lone pair of electrons also taking part in the potential of the pi electron system. So basically it is going to be have 8 pi electrons, uh, as it is cyclic in nature and planar with a 4n pi electron. See, the rule of aromaticity is 4n plus 2 pi electrons, but he is going to obey the 4n pi electrons. So it is not going to obey the 4n plus 2 pi electron system. That's why it is aromatic in nature. So I have already discussed anti-aromatic are the compound. They will give the some of the property, or they will obey some of the property of aromaticity rule, and some of the properties they will not obey. That's why they are anti-aromatic in character and they are least stable. So thus azepine and its tautomers. Now tautomers is nothing but slight structural change due to the movement of the one of the atom in the structure. So the tautomer of azepine, it is exist in the non-planar conformation, that is boat conformation. So these two are the tautomeric conformations of this azepine molecule. So one atom in this, if they are going to obey the non-planar boat conformation in which one of the atom is sp3 hybridized, that's why it is least stable. Compound do not comply. Already we have discussed that it is not going to obey the 4n plus 2 rule. It will the 4n pi electron rule. That's why azepine is non-aromatic, that is anti-aromatic in nature. Now, a tautomerism which is shown by the azepine molecule is, these are some of the tautomers of azepine molecule, in which it is 1H azepine, 3H azepine. So there is movement of the double bond across this cyclic ring. So there is formation of 1H azepine, 3H azepine, 4H azepine and 2H azepine. And the stability order of this azepine molecule is 3H is more stable than the 4H than the 1H. So the least stable atom of the azepine molecule is 1H azepine. Now, the synthetic methods of the azepine molecule. What is azepine? How it is anti-aromatic compound? This we have studied. Now we are moving towards the synthesis of azepine molecule. Now, the synthesis of azepines and their derivatives we are going to discuss in following synthetic method. So the first synthetic method for the synthesis of azepine is valence bond isomerization. Now, what is this valence bond isomerization? Why the name is given in this manner? Now, this approach for the synthesis of azepine molecule, it is used years together. So many long we have discovered this method for the synthesis of azepine molecule. Now, this reaction, it is differ mechanically from the majority of the common molecular reagent. This method of synthesis of azepine from another method, it is quite different as compared to the molecular rearrangements. Now we all know molecular rearrangement, that is during synthesis of benzene, benzoic acid, we have seen Schmidt rearrangement, Bachmann rearrangement. So the synthesis of azepine from valence bond isomerization method, it is totally different from the molecular rearrangement reaction, and it is essential for the valence bond migration. Now, in this particular method, there is nothing but only the migration of the valence bond to be take place, and there is involvement in the bond reorganization which will happen in this method. So in this method, there is migration of the valence bond happen in order to reorganize the bonds. So this reaction involves the reorganization of the sigma and pi bonds. So in this, see here, we are going to show the shifting of the bonds from one position to the another. That means there is migration and reorganization of the sigma and pi electrons and pi bonds takes place, and does not involve a migration of the any atom or group. See, in earlier statement, I told you that it is totally different from the molecular rearrangement. ज्या ज्या वेळी molecular rearrangement चा डिस्कशन येतं, in which molecular rearrangement मध्ये काय असतं, एकतर ॲटम किंवा ग्रुप ऑफ ॲटम एका पोझिशन पासून दुसऱ्या पोझिशनला मूव्ह होतात. But this is not the case with this synthesis method, that is valence bond isomerization. या synthesis method मध्ये only there is movement and migration of pi and sigma bond to be happening. Now, in this particular method, we are going to use so this method is known as valence bond isomerization method, because there is no any movement of atom or group of atom will take place. There is only the movement of sigma electrons and pi electrons will be happening. And a relevant example for the synthesis of azepine from benzene and ethyl carbonazide is a relevant example for the synthesis method of valence bond isomerization. Now, when there is a combination of reaction of ethyl carbonazide with benzene molecule will happen in the presence of light. So there is removal of this N2 molecule, and whatever this adduct is there, it is conjugated with this benzene molecule across its double bond. So when it is conjugated across the double bond, one of the double bond with this benzene molecule, so there is formation of this stable structure. This stable structure is formed because of the presence of electron withdrawing group on the nitrogen atom. So this presence of this electron withdrawing eastern functional group across this nitrogen atom, which will stabilize this ring. The parent compound, which on hydrolysis, so this stable parent compound, which is on hydrolysis in the presence of heat, will form 1H azepine, and this 1H azepine followed by isomerization, it will form a stable structure that we all know 3H azepine is more stable than the 1H azepine. So in order to that, it will isomerize itself, and it will synthesize 3H azepine molecule. So this is one of the very important method for the synthesis of azepine molecule from by which method? Valence bond isomerization method. What is that method? In this method, two simpler molecules will be conjugated together in the presence of light. So there is removal of this N across double bonds in the form of N2 gas, and this remaining adduct is conjugated across this double bond of the benzene molecule. Now this conjugation of this functional group across the double bond of benzene molecule will involve migration of pi electron and sigma electrons. Now this migration of pi and sigma electrons across the ring of the benzene molecule, during this migration there is formation of an adduct, that is this is intermediate compound, but this intermediate compound is quite stable due to the presence of this electron withdrawing group. It is attached to this nitrogen atom. Now after the withdrawing procedure of the electron, this group is removed as a byproduct through the hydrolysis reaction, and there is formation of 1H azepine, and this 1H azepine undergo isomerization procedure to synthesize 3H azepine. Now this bond reorganization, that is reorganization of sigma and pi electrons without the rearrangement of atom will be happening. So this synthesis is quite useful for the synthesis of azepine molecule. Now the second method for the synthesis of azepine is from phenylazide molecule. Now this phenylazide molecule, uh, on decomposition in boiling, so if we are going to boil this phenylazide, uh, it will decompose and to form benzazirine, and this benzazirine molecule undergo reaction with the primary amine, there is formation of a 1-azepine derivative. So it will synthesize a substituted 1-azepine derivative at its second carbon atom. And the substituted 1-azepine derivative undergo again isomerization step to synthesize a stable compound, that is 3H-azepine derivative, as it is substituted. So the name of the compound is 2-diethyl amino 3H-azepine. Now this is an important point which we are going to keep in mind, that 3H-azepine is one of the favorable stable structure of the azepine. So whenever there is formation and synthesis of azepine molecule, it is finally isomerized into a stable product, that is 3H-azepine. Now the next synthesis method of azepine molecule is from nitrobenzene. An interesting synthesis of 2-alkoxy 3H azepine. So this is an interesting method for the synthesis of azepine, that is 2-alkoxy 3H azepine has been accomplished from the nitrobenzene. So this 2-alkoxy azepine molecule we are synthesizing from the nitrobenzene by its deoxygenation with tributyl phosphine. So this nitrobenzene, it is deoxygenated in the presence of tertiary butyl phosphine, This reaction required a primary secondary alcohol. This reaction will requires the presence of alcohol, either primary alcohol or a secondary alcohol, but fails in the presence of phenol. See, very important point which we are going to keep in mind for this reaction is, that is, synthesis of azepine from the nitrobenzene molecule is, this particular synthesis requires the presence of primary or secondary alcohol, but it is absent or it will not works in the presence of phenol. So the mechanism involves an initial formation of an aryl nitride. So when the nitrobenzene molecule undergo deoxygenation in the presence of tertiary butyl phosphine, so there is formation of an aryl nitrine, and this aryl nitrine, a nitrine in the presence of either primary or secondary alcohol. So there is formation of an 2-alkoxy 3H azepine molecule will be happening. Now we are moving towards the reactions of the azepine molecule. Azepines and their derivative undergo and gives a very interesting chemical reaction, from which the first chemical reaction is thermal reaction. So if we are going to heat this N-carboethoxy 1H azepine, on heating it will result in the aromatization. As it is anti-aromatic or it is categorized into non-aromatic, it is converted after heating into an aromatic compound, that is N-phenyl carbamide. Similarly, on heating an aniline derivative, that is 2-methyl N-carboxyethyl it will also result into an formation of an aromatic compound. That means if we are going to heat this compound more than 200 degrees centigrade, it is converted into an N-phenyl carbamate, which is aromatic in nature. Now the next reaction with respect to the azepine molecule is ring contraction. Azepine undergo photo ring contraction. So this particular procedure or reaction will be happening in the presence of light. So this azepine undergo photo ring contraction to the bicyclic valence tautomer of the type shown below. So it will result into the bicyclic compound, which is an valence tautomer of this compound. These transformations are thermally reversible. So this particular reaction is thermally reversible and occur by an orbital symmetry controlled by the disrotatory electrocyclic processes. Now 2-methyl N-carbomethoxyazepine, 2-methyl N-carbomethoxyazepine undergo reaction with the light in the presence of methanol. It is converted into an bicyclic, that is 2-carbomethoxy, 3-methyl 2-azabicyclo hepta 3,6-diene. So it is converted into an compound which is bicyclic in nature in the process of 93% of the yield. So if we are contracting this with in the presence of light, it is converted into an bicyclic compound. Now, let's next important reaction with respect to the azepine molecule is Diels-Alder reaction. So this Diels-Alder reaction is nothing but formation of an cycloadduct product, and this cycloadduct product is 6 plus 2 pi electron reaction. Now what is this? This N-carboethoxyazepine cycloadds or cycloadduct with 1,5-dicarbomethoxy 3,4-diphenyl cyclopentadienone. So it is conjugated with, or it is uh, react with the 1,5-dicarbomethoxy 3,4-diphenyl cyclopentadienone, which will yield 6 plus 4 pi cycloadduct. So there is formation of an cycloadduct. It will form a cyclic compound containing 6 plus 4 carbon containing derivative. So there is formation of an this adduct by Diels-Alder reaction. Now, some of the medicinal uses of this azepine molecule. They are, it is used as an anticonvulsant. So it is used in the as an anticonvulsant, that is carbamazepine and oxamazepine. Now already you have studied the epilepsy or convulsions type of disorder if it is happening in our brain. So continuous triggering of the neurotransmitter, if it is happening in our body, it will result into the convulsion. So these azepine containing drug molecules, they can be used as an anticonvulsants. And examples of those drug molecules which can be carbamazepine and oxacarbamazepine. Then it is also used as an tricyclic antidepressant, in which there is a presence of azepine ring in its structure. So the medicinal tricyclic antidepressants are imipramine, desipramine, clomipramine, and trimipramine. Now these type of medicinal uses, they may ask for you for the fill in the blanks, for short answer questions, write down the medicinal uses of this drug. So for example, for the azepine molecule, it is used as an anticonvulsant, and it is also used as an tricyclic antidepressants. So today we have discussed a very important compound, that is azepine molecule.